Hello again, everyone. My name is Brian Barber. I'm the chair of the Canadian Forest Genetics Association and co-host of this symposium. And it is my pleasure to introduce our first keynote speaker of this symposium and our first virtual presenter in the history of the CFGA and the WFGA. One small step for us, but what must appear to be a giant leap forward to those who founded our associations back in 1937 and 55 respectively, and who used handwritten and typed letters, telegrams, and rotary telephones to organize those meetings. The assisted migration to virtual conferences also confirms that we must continue to adapt to change or perish. Dr. Nathalie Isabel joins us via cable, fiber optics, and/or satellite from her home in Quebec City. Nathalie is a research scientist for the Natural Resources Canada, an adjunct professor with the Canada Research Chair in Forest Genetics at Laval University. She's also a member of the Spruce-Up's Genomics project. Nathalie pursued a career in forestry as she wished to promote intensive silviculture and improve conditions of those working in remote locations, isolated from family and friends. Her interests have evolved, but her passion for connecting with people and nature have remained. Nathalie obtained her bachelor's, master's, and PhD from Laval University. During her master's, she spent a few months breeding edible mushrooms in Bordeaux. And as a doctorate student, she spent several months in Vancouver under the supervision of Dr. John Carlson at the UBC Michael Smith Lab. A decade later, with her family, she served as the visiting professor at the University of Lethbridge, Alberta, researching poplars. The locals were warm and welcoming. She described her experience in southern Alberta as exotic, with its cactuses, antelopes, and rattlesnakes. I too have canoed down the Bow River through Dinosaur Park, and it did conjure up images of sailing down the Nile, except lined with cottonwoods instead of palm trees. Nathalie's current research focuses on understanding the evolution, genetic diversity, and adaptability of boreal and temperate tree species. She's also part of a team working on wood identification and traceability to combat the illegal timber trade. Both aim at connecting people to forests. Nathalie also co-chaired CFGA's 2019 conference in Quebec with Dr. Patrick Lenz. They were great hosts, and remain active CFGA directors. Thank you very much. Nathalie's personal interests include mountain biking, hiking, and skiing, as connecting regularly with nature helps keep her grounded. She's also a fan of history and science fiction, and so she's therefore well qualified to present the opening keynote address, Forest Genetics and the Forests of the Future. Let's put things in perspective. Welcome, Nathalie.
Hey, thank you, Brian. So I'm going to share my screen. Okay. So I will. So, do you see my presentation?
Yes, it looks good.
Okay, perfect. So, it's a great pleasure to be here today. And I would like to thank the conference organizer, especially Brian, of course, Barber, and Nikolai Krenitz for having invited me to give this talk about the future. What a stimulating topic. But truthfully, I found it quite challenging. Well, predicting what is going to happen in forestry and how genetics will contribute to it is somewhat fictional. Fortunately, I love science fiction. Nevertheless, a way to respond to Nick's question, because actually it's Nick Ukrinitz who asked me to predict the future, is to look back and see if things have changed or not. In doing so, I've sought inspiration from Canadian painters. Actually, I think that we should see more art in scientific conference. It's really inspiring. One of these painters is Emily Carr from BC, who was born more than a century ago. She was a nonconformist. Her paintings reflect her awareness of ecological and social issues and disclose already the impact of deforestation and colonization on First Nations. I see myself a little in her story. You should have a look at it. But I have no painting, neither drawing skills. So this morning, I will present my own perspective. Yes, almost 30 years ago, at the very beginning of my career, I was a graduate student, as you are today. But most importantly, I had hopes and fears, and as you do. During those days, I was looking the group of Eric Landers working on human genome, and I was dreaming about tree genome sequencing. And you all, what are your dreams? I'd like to hear you in the next couple of days. With all this in mind, I'll share some thoughts about the achievement that have been made over the last 30 years, the challenges we are facing, and how perhaps we should envision solutions for the future. You are the main actors in this future. So what has happened since I've started a bachelor degree in forestry more than 30 years ago? Well, until the late 80s, logs were still transported from the forest to the mills on rivers in Canada. Fortunately, the face of the forest industry has changed. Not only that, but also forest management practices in general. Today, almost 50% of the forest, it looks like more than 300 million hectares of Canadian forests are certified by a third party, which means that they are sustainably managed. In the field of genetics, we have also witnessed major breakthrough. 1989, the year I started my master's degree, was also the year a group of 23 scientists set the stage for the Human Genome Project. As a result, today, for $100, you can get a good idea of your personal genetic makeup, although this also raises ethical and legal concerns. In forestry, major genome sequencing project began a decade later because of the size of the conifer genome and the fact that the research topic was rather regional compared to human, where the researcher has to deal only with a single species. In forestry, more particularly at Laval University and at the Canadian Forest Service, a forest genomic teams led by Jean Bousquet and John Mackay and supported by the province was built over time around white spruce. So in Eastern Canada, Arborea project was the first project of this kind. Of course, the focus of the project was on wood formation. And could you imagine that such a project was made possible because it built on collection of trees developed by tree breeders in the late 60s, when I was starting myself elementary school, unaware of all those efforts. Arborea and Phenomics were brought together into the Smart Forest and Spruce-Up Unified project. Our goals were to develop tools to enhance forest health and productivity, but also to better understand tree response to climate change in order to develop more resilient trees. Some activities in these two projects were specifically related or dedicated to the study of GELs. GELs mean ethical, environmental, economic, legal, and social aspect of genomics. Yes, exactly. Social licensing is an important factor to consider when the time comes to deploy material resulting from genomic selection or any other genomic tool, especially in forestry. We have to really keep this in mind. All of this has contributed to the development of application and knowledge useful to breeding and conservation program. Indeed, I encourage you to read this special issue of evolutionary application that we put together, Jason Holliday, Sally Etkin, and myself, and that was published in January 2020. you will see what has been achieved for many different tree species and what exists in terms of genomic resources. So to summarize, in forestry, even if things have started later than human, the genome of many trees have, and their associates, mycorrhizae, pathogens, insects, have been sequenced. In an approximately 20-year interval, we have moved from a vision to a reality. So my grad student's dream has come true, although I thought the result would be much easier to understand. It involves a lot of time, a lot of activities, and a constant team efforts. As you can see here, you see it's the genetic map of white spruce with the 12 chromosome, and you can see actually here the scaffold. So we have 12 chromosomes represented by a couple of millions of scaffold. So there's still a lot to do. All this would have never been possible without collaboration with numerous actors and the funding agencies. So Genome Canada and Genome Regional Center were the main funders of those projects. So what are the current and future challenges that we face? We already have an idea. We can know some of the unknowns. We know that the climate is changing, so are the forests, and as a society, we know that we will have to adapt. Not only is the economic activity resulting from forestry important, but other intangible values are being considered as well, like ecosystem services. But what will be the consequence of climate change? These are very hard to predict. Some forests will be more, the productivity of the forest will change and probably on a regional scale. Northern population of trees will be maladapted. And of course, even if we have a good knowledge of the diversity and we do assisted migration, in terms of productivity, the impact might not be very long. So forests and trees have an inherent adaptive capacity that we can describe by the standing genetic adaptive variation and plasticity that still need to be explored. However, the adaptive capacity of trees along their lifespan is not easy to capture. During the symposium, you will see various approaches, actually, and sorry, and represented on this figure and summarized in Victoria Sork's paper. These approaches are used to search for adaptive variation and quantify this variation. In one of these approaches, on the left side of the triangle, no phenotyping is required. So the objective is really to look association between genotype and environment. For less studied species, this approach, although not perfect, is very interesting because it can provide information for vulnerability assessment framework that are important for forest manager. As an example, Rose Gossen will present some of the results she obtained using a genotype environment association in aspen across its North American range. In addition, Lisa Tishenko has a complementary poster that shows how aspen sex is distributed in population across the landscape. Lynn Touchette, who as an undergrad has also contributed to this work, is here, and she did some work on the co-production knowledge. So I hope that you will find the time to chat with them. They are very inspiring. In the Spruce-Up project, we are also working actually on the other approach represented on the right side. That approach consists to determine the degree of relationship between genotype and phenotype. Most part of the time evaluated in common gardens. Unfortunately, we are really limited in terms of phenotype that can be used and to describe and measure adaptation. In forestry, we use height and diameter as a proxy for fitness. On the top of that, most of the studied functional traits consist in punctual measurement. So in Spruce-Up, we are working with ecophysiologists like Ingo Insminger and Petra Dodorico to develop precision phenotyping using drones and leaf spectral reflectance indices in relation to the physiological status of trees. It's a lot of work. As my colleague Janice Cook would say, it takes a village to do that. So this type of approach allows us to see the invisible to the naked eye. It takes few hours instead of days to assess phenology. This will allow in the near future to compare in almost real time individual following environmental stress. This is very promising, but it's still in its infancy. In the meantime, with forest ecologists, we are working on other traits that to enable the assessment of tree response to climatic constraints during their lifespan, from juvenile to mature trees. These new traits, derived from dendroecology approach, are used to assess the relationship between growth imprinted in the tree rings. Actually, tree rings are really like bioarchives, and it allows to test those variations in relation with the climate. This is quite powerful. So the different association approach were tested together in white spruce. We use the GEA and the GPA approach in common garden and with the dendroecology. So Claire Depardieu will present our latest results later today. Again, I invite you to chat with her. To summarize the known unknowns theme, we know that global change are already impacting the environment. And we know that urbanization and deforestation are just accelerating the catastrophe caused by climate anomalies that are becoming more frequent. You are already working on it. And actually the program looks amazing. So where are we gonna be in 30, 50 years from now? Well, to answer Nick's question, what will be the future? I decided to go back into the materials of the previous CFGA conference held in Quebec City just a few months before the COVID pandemic. I found this photo just amazing. It illustrates very well to me the unknown unknown. And we thought at the end of the week when that photo was taken, that we were relatively well prepared for the future, working on the known unknown. So I'm gonna take a drink. So in order to develop a vision of the future, we should have a look at the science fiction, because usually it works well. But I've realized that when we watch science fiction movies like Blade Runner 2049, Denis Villeneuve was the realizator, is a guy from near Quebec, who lives near Quebec City. So he's doing a great movie. The future looks fairly dark. In fact, all forest and plant life seem to have disappeared from the Earth's surface. Is such a scenario inevitable? And if instead of such a catastrophic scenario, we could envision a different future. Of course, we know that natural resources and water supplies might be limited and that cities will likely be home to nearly 70% of the world's population. But is it possible to imagine such a future? Of course, we need to stay pragmatic and assume that people will be vaccinated and that politicians will act. They'll do something for climate change. Why not? To this end, from the previous conference, we integrated GEL activities at the CFGA. GEL's research covers many disciplines in social science and the humanities. GEL research really offers help the bridge gaps between genomic researcher and stakeholders. So all the week during that conference, we organized many different activities. It was just great. This was the sketch of the week. Before the end of the conference, it was clear to everybody that genomics now and in the future offer a wall of possibility, and that assisted migration, industrial plantation, and fundamental research would be central themes along with forest health, and that we will still need to rely on conventional enhanced genetic programs, at least for a couple of decades. In addition, according to the survey, it appears that forest genetics and forest genomic research will likely address urban forest issue, restoration of habitats, ecosystem services in the future, and they could help also new industries to come, like pharmaceutics with metabolomics approach in the near future. In addition, a number of items were identified as challenges or barriers to forest genetic research, from research funding to inaction of policymakers and social licensing of tools and new forest practices. From our sponsor students of the time, actually, from their perspective, the major barrier is the challenge of language when we talk to each other and when we want to exchange with stakeholders. That is, the challenge of multidisciplinary integration. And we will need to work more and more with other people from outside our own specialty. So this is a huge challenge. But they propose a solution. So as a solution, one of the solutions, they propose to be that it should be part of their curriculum to develop the necessary skills needed to communicate what they are doing. They were looking for the hybrid student. And they also mentioned that there's a need to clarify our values to communicate what they are doing. They also mentioned that there's a need to clarify our values when we start such an interdisciplinary project, and that this type of project should be nurtured. So forest genetics and forests of the future, an easy answer for you, Nick, would be you are the future, the student and postdoc. You are the one that will create the future. And also, they might be. Actually, I work hard to make from them that they become a good citizen. So my take-home message are, genomics is powerful, and even more so with artificial intelligence, and this will positively contribute to society, although there are still technological challenges, especially in forestry with the phenotyping. But as we change, or as we have learned during the last year, we urgently need to address some scientific and societal challenges to maintain and perhaps increase the trust between citizen and research. Indeed, what we witnessed during the COVID pandemic may happen for any new problem or novelty. So we can be really helped by gel research, but we need more hybrid students and scientists working at the interface. We need also to better connect and co-develop tools with stakeholders, because sometimes simple solutions like the creation of novel diversity indices or certification standards that are very good to our according our vision of the problem can have negative societal impacts. We need to decolonize our approach and reconciliate with other communities. Philosophy is very useful for this, since it helps to take different posture when dealing with a particular subject. Finally, I want to say to take care of yourself. Humans are not perfect, and never forget that you are not alone. And it's fine if you feel like to be an outlier. And most importantly, don't stop dreaming. I wish you a very inspiring today's symposium, and I will please take the time to speak with people who are doing stuff with different things from yours. You will learn a lot. So good luck, and don't forget to breathe. And so I finished my question because I wanted to ask you, and I want to hear from you. When you imagine the future, what are your dreams? Thank you.
Thank you, thank you very much, Nathalie. That was wonderful and inspiring and beautiful. It's nice of you to introduce us to some of the artwork as displayed on your screen here. That is inspiring. Hopefully the students have taken that to heart and will develop their multidisciplinary approaches to forestry genetics. I encourage you to use the Q&A, the participants to use the Q&A feature on your Zoom screen, or if you're using chat, we'll get those transferred over. And I'll convey a few questions to Nathalie. And we've got about 5 minutes or so for this. So we'll just jump right in it then. First question is from our co-host, Nick Ukrainitz. We spend a lot of time and money to discover candidate genes for different traits, but these usually have little predictive power. In your opinion, what is the goal of this type of research, and what can these candidates be used for in the future?
Actually, I would say, Nick, it's we are still we are in a learning phase, and I think that it's important to continue those approach because yes, it's a learning curve, and at the end of the day, every gene is candidate for something. Perhaps from the point of view for breeders, you can see it as a lots of promise. It's lots of promise and perhaps a lot of hope, but still we have to be conscious. But we are in a learning phase. 30 years from now, people were looking at candidate genes with very few markers. Now we have thousands of genes. Yes, that money is not lost. I think that we will see later in 30 years from now that we might have discovered something, and we will be able to dig into those results. And yes, just see it as a learning phase. And we have to spend money and we have to invest if we want to learn.
Good.
I don't know if I answer your question, Nicolas.
That's good. Thank you. The next question is from Maurice Amé. At what level should hybrid students be developed? At the undergraduate or lower levels or postgraduate level?
I think that you can even from every stage. As soon as you have students who are really interested by this approach at the undergraduate level. I have one of them, like Lynn Touchette. She was there at the CFGA conference, and she really wants to do this type of work, working with communities. But she realized that she needs to better understand the genomics to be able to explain what's going to be the result, because there are many questions that are raised as soon as you talk about genomics or genetics. And for this reason, it's really important for her to do that. But in other case, I think that on my side, it came very late in my career. So I'm working, and I'm really, I feel that at the very beginning of my career, I was really interested in focusing on intragenic recombination and comparative genome mapping. But now I'm more interested to see the people are to empower the community and to make decision about the tools they may use to answer their question or their concern. So yes, it can happen. I think it's never too late to learn it.
Yeah, that's good. And if I could offer some advice too, how do you actually develop a hybrid student? I look back at my undergrad and some of the best, well, I enjoyed the forestry classes, of course, but taking social sciences and languages, really help broaden the perspective. And so that's, I would encourage students to use those your elective credits to reach out and take an art class, take a language, take some of the softer sciences as you pursue your forestry genomics degrees. Last question from Meng Lu is, hi Nathalie, do you think conifer gene function characterization using molecular assay is an important future study?
Yes, I realize that I forget to talk about the other conifer genome project. You know, in Canada, there was co-adapt3, TRIA, and co-adapt3 now, and Meng Lu is part of one of these projects. And yes, I think that we're going to do and have a look at the conifer specific gene, because we found, you know, when we did the characterization, people, when I started the very beginning of my career, I was also interested in gene conifer families, and everybody at that time, they made a real direct connection between Arabidopsis and conifer. And it's not the case. You know, those species have evolved on themselves. They have family of genes that they share, but in some cases they have totally diverged. So yes, I think it's going to be important, and it might give us some cue about some of the specific function, because trees are there for a long time. They are perennial species, and they have been there outside. You know, they have already experienced climate change before. Because like conifers, they are diverged from angiosperm more than 200 years ago, almost 300 million years ago, and still their genome is not so different from what we used to see with the dinosaurs. So it's, yes, I think that it's going to be important to continue that type of work.
Good. Well, with that, thank you very much, Nathalie. Really appreciate the time and effort you put into your presentation, and hopefully that'll inspire some students to
know perhaps a bit about Emily Carr, because you know, this is a time that, you know, she was already she was really concerned about the life of the First Nation and how things were going during those days. And I think that, you know, it takes about 100 years before something happened in Canada. So yes, things are changing, but sometimes at various scales. So Emily Carr is really inspiring, as well as Daphne Ojib. And so just have a look at those artists. They are just amazing.
Very good. Well, thank you again. We'll close it off here. The next session is you can view at your leisure. It's pre-recordings of some student presentations, postdoc presentations, operational tree improvement. But we'll go live again. John Degner, the 2015 WFJ Oral Presentation Award winner, will moderate a session with those presenters live starting at 9:25 Pacific time. So check the schedule and join you there. Thank you very much. Thank you again, Nathalie. Thank you.
And don't hesitate to contact me. I will be there.
Good. Yes. Use the chat features. That's great. Thank you.